Hello and welcome back to our COVID-19 online educational program. I hope you've had a wonderful winter or summer break and we're really glad to be back. I'm Anton Pozniak. I'm a physician at the Chelsea and Westminster Hospital and the London School of Hygiene and Tropical Medicine. I'm pleased to present this program to you. I'd now like to introduce our program directors. Here they are: Charles Boucher from the Erasmus Medical Center in the Netherlands, Dan Kuritzkes, who you'll hear more of later, at the Harvard Medical School, and Jonathan Shapiro from Sheba Medical Center. Now, I'm going to move on to the agenda, but just before that, to remind you that the program's been accredited by the European Accreditation Council for continuing medical education, so please apply for this. We've got a fantastic agenda for you, and after the break, we now need an update on where we are with virological diagnostics, and Anne-Genevieve Marcelin from France is going to tell us about this. We're then going to move on to vaccines at pandemic pace, and Kathryn Stephenson from the United States is going to talk to us about this and the latest results of clinical trials. Then we're going to get our regular update of therapeutics and clinical outcomes by Dan Kuritzkes from Harvard Medical School in the United States. And then we finish with our roundtable discussion. Now, I hope that at the end of the webinar, there'll be some really good learning objectives fulfilled, understanding the current status of vaccine development, describe the different modes of action of the various vaccines, and also understand diagnostic tools that we've got and the factors that influence especially their sensitivity and specificity, and also reflect on the diagnostic challenges we have in monitoring this COVID-19 pandemic. We'll have an update on the knowledge of therapeutic options for COVID-19 and define some of the factors that are influencing transmission rates and potentially influence also the clinical outcome. Now, please help us to improve the program by completing the survey at the end of the webinar. We're really thankful to Gilead, MSD and Janssen for supporting this independent medical education program. And also there's a special thanks to our other supporters who have helped publicize this seminar. I'd like now to move on to our first talk, which is going to be given by Anne-Genevieve Marcelin. And Anne-Genevieve is a professor of medicine at the Sorbonne University in Paris and a clinical virologist at the Pitié-Salpêtrière Hospital in Paris, France. And she's going to update us on virologic diagnostics. Anne-Genevieve, it's over to you.
Good evening. I'm very happy to be with you today to make an update on SARS-CoV-2 on virological diagnostic. I will speak about the diagnosis, and we will see the current recommendations and some alternatives. Then I will discuss the clinical significance of prolonged PCR positivity following recovery. And then I will finish by the update on seroprevalence and the use of antibody testing as a public health tool. So as you know, there are some common COVID-19 diagnostic methods. First, the detection of viral RNA. So this detection is used to detect current infection. And the primary method for COVID-19 diagnosis is based on multiple RTPCR kits that are now available. This technique can give some false negatives that may result from improper sampling or handling, or sometimes because of very low viral load. And SARS-CoV-2 RNA can be undetectable by day 14 following onset of illness in some cases. So this means that the RTPCR should be done as soon as possible after the onset of illness. Then we can use some serologic tests. They are used to detect past infection, and they provide a delayed but wider window of time for detection. They are useful for COVID-19 surveillance and the identification of convalescent plasma donors. These techniques can give some false negative. Indeed, the different platforms have various sensitivity, and there are also sometimes some false positive due to cross-reactivity, for example, with other coronaviruses. And the main question is, in case of positive, is there a immune protection if people are re-exposed? So now if we look at the IDSA algorithm for RNA testing, as you can see, for symptomatic individuals as well for asymptomatic individuals, the recommended test for a diagnosis of COVID-19 is a direct SARS-CoV-2 nucleic acid amplification testing, mainly on nasopharyngeal swabs or nasal or mid-urinate over oropharyngeal or saliva specimen. In case of negative test for people with a high suspicion for COVID-19, you should repeat the testing, and in case of low suspicion, it's not necessary to repeat the testing. So RT-PCR is the current standard test for diagnosis of COVID-19 using nasopharyngeal swabs or other respiratory tract specimens. A variety of RNA gene targets are used by different manufacturers, with most tests targeting one or more gene in the envelope, nucleocapsid, spike, RDRP, or other genes. So after specimen collection, samples undergo RNA extraction followed by a qualitative RT-PCR for target detection, and this test takes approximately 3 to 4 hours. Overall, all the RT-PCR kits that are registered perform satisfactorily with an efficiency PCR of at least 90%. On this slide, you have the results of an independent evaluation to verify the clinical performance of some RT-PCR tests. This research has been carried out in the University Hospitals of Geneva. And as you can see, on these are some examples, and you can see that the clinical sensitivity of these tests are at least 90% and sometimes 100%, and the clinical specificity is very high at 100%. So now, what do we need for more fluid and rapid diagnosis? As you know, today, the virology laboratory are overwhelmed with a lot of PCR tests. For example, in France, we are performing 1 million tests per week. So we have to find some solution to have a more fluid diagnosis. So there are some ways to simplify that. First, by simplifying the test procedure. There are some molecular point-of-care tests that can be run everywhere. There is also the virus antigen detection that can be used. Another way is to simplify the sampling procedure. And saliva, I will discuss the saliva as an alternative sample. And lastly, samples can be pooled for large-scale RT-PCR And this is a remaining question. So if we look at the rapid molecular test, they have the advantage to have a low complexity. They are rapid, giving a results within 1 hour. Most of the rapid tests evaluated used the laboratory developed RT loop mediated isothermal amplification, named the RT lamp technology. However, they are not available for clinical use in the US, and none of the European approved rapid tests are using the RT technology to date. In different studies, the sensitivity and specificity of this rapid isothermal PCR compared to the standard laboratory-based assay range from 75 to 94%, and the specificity from 99 to 100%. So we miss some studies with a robust number of patients to define the clinical sensitivity and specificity of this rapid test in comparison with the standard test on the same patients. And to date, we have no data on their feasibility in real life. So this is maybe a future option, but we need more data. In addition, the viral antigen detection could be a good option. We can detect viral antigen with some lateral flow immunoassay. As you can see that on the picture, it's very easy to run. They can be performed using swabs similar to those currently used in RTPCR, and they have the advantage to give a result in less than 15 minutes and could be used at the point of care with no need for a lab. However, these tests seem to need a high enough presence of the surface proteins to be detectable. That means that they have a chance of false negatives than PCR tests. And we would need to implement them with a clear guidance on correct interpretation. And the slides we have, you can see some evaluations of some of these antigen tests. And you can see that the sensitivity is between 60% to 89% with a good specificity. And overall in the evaluations, they show a lower sensitivity than the RT-PCR test, especially for a low viral load. So now saliva could be also a good alternative sample. And there was a study published very recently in the New England Journal of Medicine. They investigated samples from 70 hospital inpatients who had a diagnosis of COVID-19. They had for these patients nasopharyngeal swabs and also self-collected saliva from the patients. And you can see that at time of diagnosis, the first RT-PCR test, there were more SARS-CoV-2 RNA copies in the saliva specimens than in the nasopharyngeal swabs specimens. And they showed also that they found a higher percentage of saliva samples in blue than in nasopharyngeal swabs samples that were positive up to 10 days after COVID-19 diagnosis. After 11 days, however, there were more positive samples in the nasopharyngeal swabs. So saliva specimens and nasopharyngeal swabs they conclude that they have at least a similar sensitivity in the detection of SARS-CoV-2 during the course of hospitalization. In addition, they tested more than 400 asymptomatic healthcare workers with RTPCR and on nasopharyngeal and saliva samples. And they detected 13 saliva that were positive. And among them, 9 that had also a nasopharyngeal swabs, 7 were tested negative at the same time or later confirmed. So these results show that saliva have some interest. Indeed, it can be collected by the patients themselves. So it negates the need for a direct interaction between healthcare workers and patients. That is a source of testing bottlenecks and the risk of nosocomial infection. And this also alleviates demands for supplies of swabs and personal protective equipment. So given the growing need for testing, these findings provide support for the potential saliva specimens in the diagnosis of SARS-CoV-2 infection. So if we look at the kinetics of the different virological markers, you can see on this slide for the RT-PCR in nasopharyngeal swabs in blue, that in most individuals with a symptomatic COVID-19 infection, the PCR is detectable as early as day 1 of symptoms, and sometimes early between 1 and 3 days before onset of symptoms. And there is a peak within the first week of symptom onset, and then the positivity starts to decline by week 3, and subsequently becomes undetectable in most of cases. However, we have some cases in the practice of patients with a prolonged PCR positivity following recovery, and that is a very up-to-date questions. Indeed, in some studies, it has been suggested that around 15% of COVID-19 patients are retested positive for SARS-CoV-2 RNA after discharge from hospital. So why we can find this prolonged PCR positivity? Sometimes patients are discharged from the hospital when there are 2 PCR negative consecutively apart 24 hours, but it could be false negative, maybe in relationship with the sampling procedures, the quality of sampling, sample storage, or temperature and time. Another possibility is that this positive signal of viral RNA might be from the dead viruses and only viral gene fragments without active viral replication. And some cases have also reported that in some patients, we can have a delay virus clearance. For example, patients treated by glucocorticoids therapy, patients who have comorbidities or more than 65 years. So the most important question is, are these patients infectious? There are some reports from discharged patients that were retested positive in China. And in one study, the follow-up results demonstrated that there were no new infected cases from the 104 close contacts to the original patients. And in another study, no single family member were infected by the 4 recovered COVID-19 patients that were retested positive. So there is no clinical evidence of infectivity of those recovered patients with retest positive for viral RNA. There are also some in vitro data showing that there is a relationship between SARS-CoV-2 viral load and infectivity. In this study conducted in Canada, they performed the SARS-CoV-2 RTPCR on nasopharyngeal swabs, and they tried to isolate the virus from these swabs. They tested 90 samples, and they showed that the virus was successfully cultivated from 29% of samples. And it was very important to note that the positive cultures were only observed up to day 8 post-symptom onset, with a peak at 3 days. And the virus could be isolated only if our viral load was high, with a significant difference between samples with a CT in median at 18 versus 27 that were non-cultivatable samples. So there is clearly a difference between the PCR positivity and the infectivity. This results has been confirmed by this study showing that the viral culture is only rarely positive for low viral load, for CT values above 25 to 30, and after 8 to 10 days after symptom onset. And in this study, the viral culture was not positive for feces samples. So the question is now, these patients with a CT above 35, for example, are not contagious and thus could be discharged from hospital care or strict confinement for non-hospitalized patients. And this question has been recently raised by the very high impact factor journals, the New York Times, stating that maybe when you are tested positive for coronavirus, maybe you shouldn't be, because the diagnostics are so sensitive that we can face to this problem. And for example, in a lab in New York, they tested 794 among the 794 positive tests with a threshold of 40 cycles. They reanalyzed that, and for example, with a cutoff at 35, about half of those tests would no longer qualify as positive, and about 70% will no longer be judged positive if the cycles were limited to 30. So this is a very important question, and I think we have to state and to give some answer to that, because there are some implications on the contact tracing and for example. So now, if we speak about the serum prevalence, you have this very nice website. The name is SeroTracker, and you can find on this website all the seroprevalence studies that has been performed in the world. And if you click on the country, you can see the national estimates. This is a number, for example, in France, with a seroprevalence estimate between 1% and 4% among 12 studies. If we look at this website, we will see that overall the seroprevalence in the world is relatively low, although there are some difference between the populations tested. This is a very nice study from Spain that has evaluated the prevalence of SARS-CoV-2 in Spain. They tested 50,000 participants for IgG serology with a questionnaire. And this, they showed that the overall seroprevalence in Spain was at 4.6% by immunoassay. They didn't find any difference by sex, and they found a lower seroprevalence in children younger than 10 years. And yeah, as you can see here, there was some substantial geographical variability with a higher prevalence around Madrid, more than 10%, and a lower prevalence in the coastal areas, less than 3%. So in conclusion, the majority of the Spanish population is seronegative to SARS-CoV-2 infection, even in hotspots area. And these results emphasize the need for maintaining public health measures to avoid a new epidemic wave. So these are some sero-survey, but there is an important point because some studies now have shown that there is a decline of the humoral response over time. Indeed, in this study, they tested IgM and IgG 8 weeks after exposure. So they tested people at time of for serology at time of acute phase and then at the convalescent phase. And they showed that 40% of asymptomatic individuals, IgG plus at the acute phase became seronegative, and 13% of the symptomatic became negative at time of convalescent phase. So there is clearly a decline of the seroprevalence over time, and they show that similar observations for the neutralizing antibody, with a decline of the neutralizing antibodies over time, as well in the asymptomatic, as well in the symptomatic patients. So in conclusion, the relatively low seroprevalence and its decrease within 2 to 3 months after infections highlight the potential limits of serology for a diagnostic and the need of timely serosurvey. And this can have some implication because, as you may know, there is a question about the possibility of reinfection. And there was recently a case report of a 33-year-old man, apparently immunocompetent, who lived in Hong Kong. And this patient had in March 2020, symptomatic SARS-CoV-2 infection with cough and sputum, sore throat, fever and headache for 3 days. He had a PCR positive on nasopharyngeal swabs, and he was discharged after 2 consecutive negative PCR. At this time, he was in IgG negative at day 10. And so several months after, he traveled in Europe and came back to Hong Kong. And then he was tested at the airport. He was totally asymptomatic, but he had a PCR positive. And during the exploration, he had also an elevated CRP, and he became seropositive at day 5. And it was very interesting because they compared the whole genome sequencing of the virus between March 2020 and August 2020. And this is clearly a different virus, a different clade virus. So this case showed that reinfection, this case was probably a reinfection instead of a persistent viral shedding from first infection. So in conclusions, for the diagnostic part, RTPCR is a recommended test for diagnosis of COVID-19 in symptomatic and close contacts for test, track, and trace. This test has to be done on nasopharyngeal swabs, and there is clearly a growing need for testing, so we should find some alternative tests, for example, RT-LAMP, antigen detection, or some alternative samples, for example, saliva, that seems to be a good alternative. And I think we have to make the distinction between diagnostic and large-scale screening, because for large-scale screening, I think we can tolerate some tests with less sensitivity if these tests are more convenient and rapid. However, for diagnostic, we really need a very sensitive test, such as RT-PCR. But these different kind of tests could be very complementary. In conclusion, for the clinical significance of prolonged PCR positivity following recovery, studies shown that it can be fine in around 15%. However, there are no clinical or in vitro evidence of infectivity of these patients with a very low viral load and very high CT. And probably these patients could be discharged from the hospital care and strict confinement for non-hospitalized patients. However, there are some particular cases for immunocompromised patients that could be different. And to finish, for the use of antibody testing as a public health tool, indeed, serology epidemiological studies are very useful to evaluate the circulation of the virus and the herd immunity. However, there are some limits. I showed you that there is a decrease of seroprevalence and neutralizing antibodies within 2 to 3 months after infection. So we really need to be very careful for the interpretation of these studies, and we need a timely serosurvey. This implies also that there is a risk of reinfection, although the description of cases is very rare to date. And this emphasizes the fact that we need absolutely to maintain the public health measures. So thank you very much for your attention, and I will be very happy to answer to your question.
Thank you so much, Anne-Genevieve. A fantastic talk. And we're going to have some questions at the round table. So for you guys online, please get to the Q&A and ask your questions. I'll warn you in advance, Anne-Genevieve, we've had a lot about the cycling threshold and the false positivity rate. So I think we might start off the round table with that question. But before we get there, it's with great pleasure I introduce Dr. Kathryn Stephenson, who's a physician scientist. She specializes in infectious disease and HIV immunology. She's the director for the Center for Virology and Vaccine Research at the Clinical Trials Unit of the Beth Israel Deaconess Hospital in Boston. And she's one of these people that translates basic science into phase 1, phase 2 studies. Kathryn, the screen is yours. Hello.
Today I'm going to talk about COVID-19 vaccines, which have been developing at a pandemic pace. And I'm going to focus my talk today on the latest results from clinical trials. I don't have any conflicts of interest, personal conflicts of interest to discuss. So I'm going to start by talking about natural immune responses to SARS-CoV-2. Because all vaccine development starts with understanding what our natural immunity is. So in this particular study, 149 individuals who had recovered from COVID-19 were looked at. And what these investigators found was that there was a wide range in the immune response that developed in these individuals, with in particular a wide range in their neutralizing antibody responses. These responses are shown here in this figure on the right, which stratified them by those that patients that remained outpatient and those that had been hospitalized. So here they found that about a third of people who recovered from COVID-19 had pretty low titers, whereas there were also a couple of individuals that had extremely high titers. So in general, it's a wide range. These investigators also did a deep dive into those antibodies to see what they really looked like. And what they found is that the antibodies from different individuals were actually quite similar to each other. They all targeted the spike protein, which is the protein on the surface of the virus. And they also targeted the same actual epitopes of that spike protein. In addition, even if they found them at only low levels, they did find potent neutralizing antibodies in all of the individuals that they looked at. This is really important for vaccine development because it supports the concept that a vaccine can work in a broad spectrum of individuals. It means that all of us can potentially make a neutralizing antibody response, which we think is critical for protection. There's some additional data emerging about the durability of these immune responses. This is a study that was done in Iceland, was published this month. They looked at over 1,000 individuals who had COVID-19. And they looked at them looking at these antibody responses over time, and that's shown here in this figure. Again, they stratified those participants by those that had been hospitalized and then over the time that they had recovered. So they found that 91% of the patients they looked at were seropositive, meaning they developed antibodies by 25 days, and that those antibody titers increased over the 2-month period and then stayed steady. What they found at 4 months of follow-up is that there was no significant decrease in those antibody titers. Hospitalized persons seroconverted more frequently and quickly, and antibody titers were most strongly associated with hospitalization and clinical severity. But what's most important for us in terms of vaccines is that this data provides evidence that antibodies can persist at least over a 4-month period. We don't know too much yet about whether antibody responses in humans actually protect us from reinfection, but we have looked at this question a little bit in non-human primates. So this is a study that came from the group where I'm based, the Center for Virology and Vaccine Research in Boston. So what my colleagues did in this experiment is they had 9 rhesus macaques, and they were asking the question if animals can get reinfected with COVID-19, meaning if they got infected once and then recovered, could they be infected again? So what they found first is that when they challenged these monkeys, which is shown here, that they, the first time, they all had high viral load in their lungs. And this peaked in the first few days and then came down quickly over a week and then fully resolved by 3 weeks. These animals, after this challenge, all recovered and developed antibodies, neutralizing antibodies. At day 35, so not too much long afterwards, the animals were re-challenged again. This time, this is shown in the figure here, was a very different response. The second time, animals, only 3 of them had low viral load in the lungs with no recoverable live virus after at all. And no viral DNA was detected in the lungs at any other time point past the first day. And these animals all had a rapid boost in their immune response, showing immune memory. So this data is really showing that natural immunity is possible, and at least it was demonstrated in monkeys. Our group has also been able to show, along with many others at this point, that vaccines can induce this immunity, and it's also protective, at least in animal models. So in this experiment, my colleagues used an adenovirus 26-based vaccine. That's a viral vector that expresses the full-length spike protein. They actually immunized 52 monkeys with some different variations of the spike protein. And then again, they had some that got placebo. These animals all developed neutralizing antibody titers, and the best vaccine was the one that expressed the full-length spike protein. This particular vaccine that I'm showing here is the one that's progressing in clinical development. The antibody titers in these animals were quite high. They were actually 4-fold higher than what was observed in convalescent humans. So it's possible we might be able to do better than what's seen in humans with vaccines. And these monkeys had nearly complete protection after they were challenged. So here's the animals that got the placebo. They all had virus in their lungs. And here are the animals that got vaccinated with the best vaccine. and they did not have virus in their lungs. In addition, it looked like neutralizing antibody titers inversely correlate with protection. And this is also shown here where we separated out the animals by those that were completely protected versus partially protected for animals that got the different vaccines that weren't as effective. And this is showing that neutralizing antibodies are a correlate of protection and may be a mechanism of protection. A follow-up study also showed that these vaccines can protect animals against severe disease as well. So now I'm going to move on to the clinical data. So there are a lot of different vaccines that are in clinical development, and it would take days to go through them all. But what is important to know is that those that are in advanced clinical testing all share the same spike protein. They all express spike protein. It's just different mechanisms of delivery. Today, I'm going to focus on 6 vaccines. These are ones that are in phase 3 efficacy testing, but that also have published clinical data. And here are the 6 vaccines I'm going to review today. For this talk, I'm going to refer to them by the pharmaceutical companies that are sponsoring these trials and producing these vaccines, though most of these vaccines are developed in collaboration with academic groups and also government groups. If anyone is interested in doing a deeper dive or just checking in on their favorite vaccine, I recommend going to the New York Times, which has a coronavirus vaccine tracker. And that will tell you about the entire pipeline of vaccines. So I'm going to start by talking about the Pfizer vaccine, which may be the most developed and advanced in development, at least in Europe and the United States. So this vaccine is a lipid nanoparticle that encapsulates a modified RNA that expresses the spike protein. Now, they haven't published this data in a peer-reviewed journal, but they did make it available in medRxiv. And they put up their phase 1 data. So what they did in this phase 1 study is they did a randomized controlled trial, and they had about 90 people who received or were in the arm that got this BNT162b2 vaccine. The study was actually much larger than that. It looked at additional vaccine, but today I'm just going to tell you about this one because this is the one in phase 3 testing. So they looked at 2 vaccinations 21 days apart, and they looked at 3 different doses. And they also looked in young people, 18 to 55, and then an older group, 65 to 85. So what I'm going to do for this vaccine and all the others is I'm just going to tell you first about what was reported for vaccine safety and then what was reported for the immune response. So here the vaccine safety data show that there were no serious adverse events and no study pauses, but strong big caveat that they reported only 7 days of follow-up so far from their second vaccine, so not a lot of time. The symptoms that they observed were relatively mild. You can see here that the majority of participants actually had no symptoms at all, and really fevers were very uncommon following immunization. Symptoms were somewhat dose dependent. Here's the 30 microgram dose, and they were higher after the second dose. They actually went up with the second dose. But in general, it was very well tolerated. The antibody responses for this vaccine were quite good. So anti-spike antibodies were generated in all of the participants after the first dose. But for really good immune response and neutralizing activity, it really required 2 doses. So in this figure, you can see these small bars are showing the responses after the first dose, and then these big bars are after this boost. And then the immune responses were seeing the same pattern in both the younger adults and the older adults. The lowest response was with the 10 microgram group. And importantly, even in the older adults, the titers were as high as convalescent plasma, which is shown here, convalescent serum, and actually were higher, significantly higher for the younger adults. And they didn't have any data to share yet for their T-cell responses. So the sister study to Pfizer, or sister platform, is the Mederna vaccine. And this is also a nanoparticle that also encapsulates an mRNA. So very similar concepts. Here they've published their phase 1 data in the New England Journal. This was done a couple of months ago. Now the study design for their phase 1 was an open label trial with 45 participants, 2 vaccinations, 28 days apart with 3 different doses. These are some higher doses than what was tested in Pfizer. So here they found no serious adverse events and no study pauses. And after the first dose, symptoms were relatively mild, and probably a majority of participants didn't even have systemic symptoms. But after the second dose, it was quite interesting. There was a big jump in their systemic symptoms. You can see here that 100% of participants at the higher doses had systemic symptoms. And actually 21% of those at the highest dose had severe symptoms, which in a clinical trial means that it really interfered significantly with their daily life, and required, in a lot of cases, required some sort of medical treatment or intervention. These severe symptoms were essentially a febrile syndrome, so fever, malaise, chills. And here, about half the participants had fever, sometimes as high as 103. Their immune data was great too, similar to Pfizer. So anti-spike antibodies were generated in all of the participants. And neutralizing antibodies, again, required 2 doses. This is for the high dose. You can see that there's a little bump in immune responses, but really after the boost, a big bump. The lowest responses were with that low dose. And what's really cool in terms of their immune responses is that they were really at the higher end of what's seen with convalescent sera. T-cell responses were characterized in this study. CD4 T-cell responses were detected, and they were strongly biased towards what we would call a Th1 phenotype. And this is relevant because we are looking at the risk for vaccine enhancement of disease, which is typically associated with a Th2 biased result. So this is supportive that these would be safe. So moving on from the 2 RNA vaccines, I'm then going to discuss the AstraZeneca vaccine, and this is a different platform altogether. This is a viral vector vaccine. It's based on a chimp adenovirus vector, again, expressing spike protein. This paper came out in Lancet. It actually summarizes about 1,000 participants' data, so quite a bigger study. And here it was an interesting design. They looked at 5 × 10⁻⁻⁻ viral particles, basically 1 shot of this, and almost all the participants, it was just 1 dose. And they didn't compare it to saline. They compared it to a meningitis vaccine, which has its own little profile of reactogenicity. They also had 1 arm of the participants that received prophylactic paracetamol or antipyretic, meaning before they got the vaccine, they took that dose. In terms of vaccine safety, there were no serious adverse events and no study pauses in this phase 1/2 study. This was a much more reactogenic or symptomatic vaccine than what I reported earlier, at least in terms of after the first dose. So after just 1 dose, systemic symptoms were quite common. You see them here. It's this febrile syndrome, fever, chills, joint pain, malaise. About 60% of participants experienced that, and there was quite some high fevers. So 2% of participants had temperatures over 102. Yes, taking antipyretics did prevent many of those symptoms, but it's unknown fully whether that would affect immunogenicity. Here, in contrast to the RNA-based vaccines, symptoms after the second dose were actually less severe. So they had 10 participants that they boosted, so not a lot, but in that small number of people, the symptoms seemed to go down the second time. The immune data, interestingly, was, to my eye, not quite as good as what was seen with the mRNA vaccines. Here, anti-spike antibodies peaked at about a month. Now, remember, most participants only got 1 shot. And neutralizing antibody titers were relatively low after that single dose. So granted, I'm comparing kind of apples and oranges because we're looking at 1 dose versus 2, but the 1 dose results were really at the bottom of what would be seen in convalescent plasma. In the 10 individuals that got boosted, those responses did go up, which is great, and they were more comparable to convalescent samples, though not necessarily at the high end. So I think this is showing that for this vaccine, I think a single dose is not going to be enough. T-cell responses were detected, but they didn't report on Th1 or Th2 phenotypes. So now I'm going to talk about Novavax, and Technically, this phase 3 study hasn't started yet, so it's supposed to start in a couple of weeks, but they did publish their phase 1 data in the New England Journal, and for that reason, I wanted to share it with you. So this is also a nanoparticle-based vaccine. So this is similar technology to the mRNAs, but instead of encapsulating the RNA sequence, it encapsulates the spike protein itself. And this was administered with or without an adjuvant. This was a randomized placebo-controlled trial, which they conducted in Australia. They looked at 2 doses. They looked at 1 or 2 administrations of that, and then again, with or without this adjuvant. In terms of vaccine safety, this was a pretty well-tolerated vaccine. There were no serious adverse events, no study pauses. And the majority of individuals, I mean, looked at after the first dose, really the majority had no symptoms, or it was very similar to placebo. Only 1 participant in the whole trial after any of the doses had a fever. There was maybe slightly more symptoms after the second, but still very well tolerated. And the antibody responses were also quite good. So a single shot did elicit binding antibodies in nearly everyone, but the boost really did make those antibodies jump. The adjuvanted 2-dose regimens were the most immunogenic, 2 doses with adjuvant, and that it really didn't make a difference at the highest doses, whether or not they both performed similarly. So here is 5 micrograms adjuvanted, 25 micrograms adjuvanted. You can see how the boost really raises up those values. And these high values that were observed were actually 4 to 6 times greater than matching convalescent serum. This is a contrast, I think, to the chimp adenovirus vaccine. Granted, if you recall, that was only 1 dose. Okay, so I'm going to end by talking about 2 vaccine programs that are outside of the US and Europe. And the first is the Ad5 vector vaccine program in China, which is the CanSino vaccine. This is a replication defective Ad5 vector. They were actually the very first to publish human data. And in fact, they're the only ones that I can see that have 2 separate peer-reviewed publications for their phase 1 and their phase 2 data. So they're really on top of it. Their phase 1 trial was an open label study in Wuhan. Phase 2 was randomized controlled in Wuhan. In the first study, they found, they tested 3 doses, and they found that the high dose was really not well tolerated. This kind of febrile syndrome was seen, and the neutralizing antibody titers after a single shot were not fantastic. They were low and moderate. The phase 2 study, it was interesting. They dropped the high dose and went with just low and middle, but here they found that even the middle dose was not well tolerated. Actually, 9% of the participants had severe adverse events, mostly with those temperatures higher than 101.3. So it's in Celsius, getting around 39 in Celsius. And the low dose is immunogenic as the high dose. That's seen here. But to my eye, these neutralizing antibody titers are not terribly high, and they don't compare them to convalescent sera in this paper, but I think they're probably lower than observed with the other vaccines. But again, 1 shot. So if it's 1 shot, you know, who knows, maybe a second shot would boost it. Phase 3 trials are underway in China and the Middle East, but I mentioned here that there's actually limited approval for this vaccine in China, where they recommended it or are administering it to Chinese military, but it's unclear the details of that. The last vaccine constructs I want to talk about is the controversial Russian vaccines. Now, what they've reported is that this was an Ad26 prime followed by an Ad5 boost expressing spike proteins. They did publish some data in Lancet just a couple weeks ago, which they call a phase 1/2 study, though I have to say that the sample sizes are quite small to be really considered phase 2. What they looked at, both of these are open label trials, and the vaccine combination, which is what's going into clinical use, they really only reported data on a total of 40 participants between the 2 studies. What they reported is that, consistent with prior studies of these vectors, they were generally well tolerated. About 50% did have fever, so this seems to be common with these vector vaccines. And then the neutralizing antibody titers look to be, here you can see them here, they look to be comparable to convalescent plasma. So not those 4 times as high and not low, but like right in the middle. So the phase 3 study is apparently underway, but there's not a lot of details. What makes it very controversial is that it was supposedly approved in Russia and recommended for use, but so far it's actually unclear what the uptake for this is. And I just want to kind of point out again that this is all very, very small sample size for a safety cohort. So there's really not a lot we can learn from this, even from this published paper. So now I'm going to summarize what I've told you today. So COVID-19 leads to neutralizing antibody responses in humans. There's a wide range in the titers. It's likely related to the severity of illness. Neutralizing antibodies target the spike protein, but it's unknown at this moment, the data is developing, if these neutralizing antibody titers are actually protective in humans. Animal studies suggest that immunity to COVID-19 is possible. This is shown with convalescent neutralizing antibodies that were protective against reinfection, and also that vaccines can protect animals from COVID-19 infection and severe disease. Nearly all of the COVID vaccines that are in development use the spike protein. It's just the delivery mechanism that is different. Finally, phase 1 and 2 clinical data really are promising. Multiple platforms were shown to be safe and generally well tolerated, depending on the doses. The vaccines all elicited antibody responses of one kind or another. There's definitely room for improvement. Some participants had a fairly severe but transient febrile syndrome that was kind of dose limiting in a couple of these studies. And also really all of them showed that 2 doses appear to be required, at least with these platforms, for a strong neutralizing antibody response. So the single shot for these vaccines didn't seem to do enough. And then finally, I want to remind everyone that the durability and the protective efficacy is really currently unknown for all of the COVID-19 vaccines at this time. So none, I have not seen any data yet that showed for sure that these vaccines are effective at preventing COVID-19. But hopefully we will see that data in the months to come.
Thanks, Kathryn, for that. That was really interesting, and especially the updates on the vaccines. I was particularly interested in your also the review of the Russian vaccine, which got a lot of press. I still am a little bit anxious about vaccines, and we'll talk about my anxieties in the session, the round table, because I think if COVID stays with us forever and doesn't disappear, we do need durable responses if we're going to go down the vaccine route. But anyway, we're going to move on to our next talk now, and it's with great pleasure I introduce Professor Daniel Kuritzkes. He's a Harriet Ryan Albee Professor of Medicine at Harvard Medical School, and he's Chief of the Division of Infectious Diseases at Brigham and Women's in Boston, Massachusetts. And Dan is not only an expert in HIV therapeutics, but an expert in COVID-19 therapeutics, and he's been giving us regular updates in these webinars, and I really look forward to this talk. Thanks very much, Dan.
Hello, I'm Dr. Daniel Kuritzkes, and today I'll be talking about clinical outcomes and therapeutics of COVID-19. These are my disclosures. I'm going to start with a clinical update since we're now 9 months into the COVID-19 epidemic, and it's a good time to take stock of what we've learned about the epidemic in terms of its epidemiology and risks for mortality in order to set the stage for my talk on therapeutics. This slide shows the cumulative incidence of COVID-19 cases and deaths as of September 9th. You can see that there were just short of 28 million reported cases of COVID-19 across the world, with more than 900,000 deaths, and in the United States alone, more than 190,000 deaths, with Brazil, India, and Mexico coming close behind. The United States has actually experienced 2 peaks in COVID-19. The first occurred earlier in the spring, as shown here, with a peak occurring at the very end of April. And we were misled by thinking that the incidence of COVID-19 was declining as cases began to drop. But this drop in cases was really driven by control measures in the states most severely affected at the time, particularly in New York, in my own state of Massachusetts. As you can see in these dashed lines that I've added, what was really happening was a dramatic decline in the northeastern part of the United States that masked a rapid rise in cases in other parts of the United States, particularly in the southeast and in the Midwest, and which led to the subsequent second peak that occurred throughout the summer and has now been falling off, but with many concerns that we are reaching a plateau of about 40,000 cases per day. In addition, we saw in the United States a huge peak in deaths in March, April, and May, reaching approximately 100,000 deaths by the end of May. But we've now had nearly another 100,000 deaths through the middle of September, with projections that we might reach 300,000 deaths by the end of the calendar year. Deaths have not been solely due to COVID-19. This slide shows the excess mortality that has been experienced during the months of March and April, during the period from March through June of this year, where the dashed line shows the expected mortality for the same period based on previous year's data. The solid black line shows actual reported deaths from all causes during the same period of this year. And this sort of slate or blue-gray color indicates the numbers of deaths due to COVID-19. This slim brownish orange line here represents cases suspected of being COVID-19 but not diagnosed. But you notice there's this big tan area, which is additional excess mortality not due to COVID-19 itself, suggesting an effect on death due to other medical conditions that could not be cared for appropriately due to the strain on the healthcare system. This slide just focused just on the top line here, gives some actual numbers, and here we are speaking just about the period from March to April. You can see that it would be expected to have about 420,000 deaths during this 2-month period in a typical year. And there were, in fact, more than 505,000 deaths, an excess mortality of 87,000. Only 56,000 of those were due to documented COVID-19, with another nearly 31,000 deaths either due to undiagnosed COVID-19 or more likely other causes, again, because people with heart disease, kidney disease, lung disease could not get the care that they needed in hospitals that were straining to take care of all of the patients with COVID-19. This study, now published in Nature from England, shows a multivariate analysis that analyzes a variety of risk factors, and what emerges overwhelmingly as the most important risk for mortality is age. Compared to individuals who are in their 50s, who serve as the reference population, you can see that each additional decade of age carries a substantially increased hazard of death, with those in their 80s having a more than 10-fold greater hazard of death compared to people in their 50s, and those in their 40s being relatively protected. Other important medical conditions that contribute to the risk of mortality include severe obesity, deprivation, malignancy, diabetes, and of course cardiovascular disease and pulmonary disease that are shown in the rest of this extensive tree diagram. Another notable factor that emerged in these analyses, and this is now from data in the United States, a study led by my colleague David Leaf from the nephrology division at Brigham and Women's Hospital, analyzed mortality in approximately 2,200 patients admitted to the intensive care unit with COVID-19 at 65 different hospitals across the country, and found that hospitals that took care of a larger number of patients with COVID-19 and had more ICU beds, had a much lower risk of mortality than smaller hospitals with fewer ICU beds. I think this can be explained by two factors. One, experience counts. The more experience providers have in caring for patients with COVID, the better they get at providing that care. And secondly, size matters. The larger academic medical centers and other hospital centers that have a deep pool of personnel resources, whether it's residents and fellows or other attending staff and nursing staff that they can call on, clearly helped to mobilize resources that may have been unavailable at smaller community hospitals to cope with this pandemic. Well, let's turn now to talk about therapeutics per se. I'll cover three topics: remdesivir, corticosteroids, and convalescent plasma. Speaking first about remdesivir, this drug is an RNA-dependent RNA polymerase inhibitor with wide activity against RNA viruses, including against Ebola, SARS, MERS, and of course, SARS-CoV-2. In vitro studies show submicromolar EC50 activity against SARS-CoV-2 in cell lines and in respiratory epithelial cells. And animal models in the non-human primate have shown the drug to be effective against experimental infection with SARS-CoV-2. The first randomized clinical trial to demonstrate the benefit of remdesivir as compared to placebo was the ACT-1 study, which was a double-blind trial in hospitalized adults with COVID-19 pneumonia. Participants were randomized evenly between remdesivir or placebo, with the primary endpoint being the time to recovery within 28 days of hospital admission. This preliminary analysis was conducted after slightly more than 600 recoveries were attained, and you can see that overall, the participants who received remdesivir had a faster time to recovery compared to those who received placebo, and this was highly statistically significant. This advantage was most notable for patients who were receiving oxygen at the time that they entered into the study. There was less of an advantage for those who were not yet receiving oxygen, and really no difference in outcome for those who required either high flow oxygen or were already receiving mechanical ventilation. Remdesivir is relatively well tolerated. There was really no substantial difference in the most common adverse events seen between the remdesivir group and the placebo group in this trial. Now, two additional studies which were conducted by Gilead, the manufacturer of remdesivir, are the pair of simple trials, one done in severe disease and one done in moderate disease. The severe disease trial enrolled participants who had significant oxygen requirement and oxygen saturation on room air that was less than 94%. These individuals all received remdesivir, so there was no placebo control. The comparison here is a 5-day versus a 10-day course of the drug. And you can see that overall, although there were some numerical differences, these differences ended up not being statistically significant, and there was no difference in mortality. If we drill down into some of the more specific outcome metrics, and these metrics are now commonly used in studies of COVID-19 using an ordinal scale that characterizes participants in terms of their need for supplemental oxygen. Now you can see that people who entered the study needing invasive mechanical ventilation, many still ended up on a ventilatory support at the end of 14 days. Those who entered with the lower oxygen requirements did comparatively better, but there really aren't any differences between the 5- and 10-day groups. This study was also used then for another analysis that compared participants in the trial who received remdesivir to a comparison group that was enrolled contemporaneously at centers that were not participating in the actual drug trial, and found that there was a reduced mortality in the remdesivir group compared to the non-remdesivir group. That is, the recovery was about 75% in the remdesivir group compared to just under 60% in the non-remdesivir group, with 12.5% mortality compared to 7.5% in the remdesivir group. Now, it's important to emphasize this is not a randomized comparison. This is not a true placebo control group. It's a contemporaneous control group. The simple trial in moderate disease was a 3-arm double-blind study that did include a placebo. Again, had a comparison of 5 versus 10 days of remdesivir, and with a comparison of standard to care. Entry criteria here were an oxygen saturation better than 94%, and of course, evidence of SARS-CoV-2 infection. This trial initially enrolled 600 participants with a primary endpoint of clinical status at day 11, and overall, the 5-day arm showed a significantly greater clinical improvement as compared to standard of care, which was statistically significant. This slide, using that same ordinal scale, shows here compared to standard of care that the 5-day group had a statistically significantly better outcome in terms of all of the different ways of characterizing the study participants. This difference was not statistically significant for the longer treatment. However, at day 14, both the 10-day and 5-day courses showed the same statistically significant improvement. And then at day 28, the 10-day course still shows a statistically significant improvement, and the 5-day arm just misses being statistically significant. So overall, a clear significant advantage for remdesivir compared to standard of care. Well, let's talk now about corticosteroids. The reason to think about using steroids and other immune response modifiers in COVID-19 is that although the early stages of infection are dominated by viral infection per se and the viral destruction of respiratory epithelial cells, this sets the stage for a second pulmonary phase and later hyperinflammatory phase that's really predominated by the host immune response to the virus and subsequent immune-mediated lung injury. The recovery trial is a large randomized placebo-controlled trial of several different possible treatments for patients admitted to hospital with COVID-19. And more than 11,500 participants have been randomized to the various arms of that trial, several of which have now been completed and reported out. The hydroxychloroquine study had a negative result, as did the lopinavir-ritonavir trial. And I will talk about the positive results from the dexamethasone study. Several of the other arms are still ongoing. This slide shows the results of the dexamethasone study, which randomized, as you can see here, more than 4,000 participants to standard of care, which was a shared control arm across all the arms, and more than 2,100 participants to dexamethasone. The individuals received 6 milligrams a day of dexamethasone for 10 days. And what you can see is that overall, there was a statistically significant reduction in risk of death due to COVID-19 in the dexamethasone group. That advantage was most pronounced in participants who were sickest, those requiring invasive mechanical ventilation, and was also significant among those who required supplemental oxygen. But no benefit was observed, and the potential for harm, although not statistically significant, observed in those receiving dexamethasone who had earlier stage disease and not yet requiring supplemental oxygen. Now, a meta-analysis has been conducted of numerous studies of various corticosteroids, all of which were underway at the same time as the recovery trial, and these other studies ended up ending prematurely because of the convincing positive results seen from the recovery, and then the investigators banded together and undertook a meta-analysis that was published recently in JAMA and was really led by the WHO and forms the basis for WHO guidelines around the use of corticosteroids. What you can see here in this tree diagram is that all of the studies with the possibly, and all of the people in all of the different risk categories show benefit of steroids. This included those receiving mechanical ventilation and those not yet ventilated, those receiving supplemental oxygen coming predominantly from the recovery trial. There may be some question about the role of vasoactive medications in this setting, but the numbers here are a bit small. Clearly a benefit regardless of age or gender, and whether people receive treatment within 7 days or after more than 7 days of symptoms. These data in aggregate have led to the WHO guidance on corticosteroids for COVID-19. The first recommendation being that systemic corticosteroids, whether dexamethasone or others such as hydrocortisone or prednisolone, should be used in the treatment of patients with severe or critical COVID-19, and this is a strong recommendation. In addition, there's a recommendation not to use systemic corticosteroids in the treatment of patients with non-severe COVID-19. That is a conditional recommendation. The evidence on which the panel bases this recommendation is with a moderate certainty of evidence is the mortality reduction of 8.7% and 6.7% in COVID patients who are critically or severely ill, respectively. Key practical issues for the use of systemic corticosteroids are that the dosing would be 6 milligrams daily of oral or IV dexamethasone, or 50 milligrams intravenously every 8 hours of hydroxycortisone for a 7 to 10 day period. Let me turn now briefly to a discussion of convalescent plasma. Convalescent plasma from survivors of COVID-19 infection may contain high titers of neutralizing antibody, and this approach to treatment has shown some benefit in managing avian influenza, H1 influenza, and MERS. Pilot studies and uncontrolled studies have suggested a possible benefit of convalescent plasma in COVID-19. A phase 2 open-label randomized clinical trial of convalescent plasma conducted in China at 7 medical centers in Wuhan enrolled 103 participants who were randomized evenly to convalescent plasma or standard of care, where the units of plasma were screened for spike protein specific IgG titers directed against the receptor binding domain that were 1 in 640 or greater, with a primary endpoint to time to clinical improvement within 28 days. In this small study, which is really a phase 2 trial, as I mentioned, there was no advantage of convalescent plasma overall, but in those with severe disease, there did seem to be a statistically significant benefit. And by contrast, in those with life-threatening disease, the sickest participants who were already intubated, there was no advantage. Joyner and colleagues have published in preprint form their experience in the expanded access program that was run from the Mayo Clinic in Rochester, Minnesota. They included several different analyses, which I'll just briefly summarize here. Looking at the 7-day adjusted mortality, here analyzed on the basis of whether the units of plasma had low, medium, or high titers of antibody, not necessarily neutralizing any, but just antibody as measured by an IgG assay. There appears to be a trend towards a greater benefit, a reduction in mortality among those who receive the high dose or high titer plasmas, and that trend is still seen in the 30-day adjusted mortality, although these differences are not statistically significant. However, if you then also stratify on the basis of whether participants or recipients received plasma within 3 days of admission or more than 4 days or later, there may be a benefit in the high dose group for those who got the earliest doses of drug or plasma compared to those who received later. Again, just missing statistical significance. So I think the issue around convalescent plasma is that we still don't really have firm placebo-controlled data for a benefit, and we desperately need the results of the ongoing placebo-controlled trials. So let me end with this summary. The COVID-19 pandemic has resulted in substantial excess deaths in the United States and around the world. Remdesivir reduces time to recovery in patients with moderate and severe disease. Its use in early disease is currently being explored. Corticosteroids improve survival in patients with COVID-19 who require supplemental oxygen or mechanical ventilation, but should be used with caution in patients with earlier stages of disease. Convalescent plasma may provide benefit in patients with severe COVID-19, but we're still awaiting data from controlled trials. And at the same time, studies are just getting underway with much more specific approaches using monoclonal antibodies directed against the spike protein. And we eagerly await the results of those trials as well. So thanks for your attention, and I'll turn this back over to our moderator.
Thanks, Dan. That was excellent and really great review of what's going on. I hope everyone can now come online for the panel discussion. But while that happens, Dan, it all seems to happen to you that between recording this and a day later, we have some new data, and there was some data on baricitinib with remdesivir. And I don't know if you'd like to just, well, we've only heard from a press release, but I don't know what your initial thoughts are on that.
Sure. So I showed the results of the ACT-1 study, which was a randomized study of remdesivir versus standard of care. The ACT-2 study combined remdesivir with baricitinib or control, and so asked whether additive therapy was better. And from what we've seen in the press release, and we don't really have any formal report from the trial yet, there seems to be a modest but statistically significant benefit of adding baricitinib, which is one of the kinase inhibitors, in order to reduce inflammation. Participants who received the combination of remdesivir and baricitinib on average recovered 1 day more rapidly, were discharged from hospital one day sooner. What the downsides of baricitinib in terms of side effect profile might have been, and exactly what are the characteristics of the participants who had the greatest benefit remain to be seen in a full report. And so just how you might add baricitinib, which is already approved for other indications, to remdesivir is very much an open question.
Yeah, I mean, it comes down to the timing of these drugs. Do you think that that's crucial? It seemed to me that the stage at which you're at is really important for those therapies, and maybe the baricitinib was given to those, more to those patients who were further on.
Yeah, well, it could be. As I said, we'll really need to see the full study report. We do know that data from other trials of immune modulators such as tocilizumab and sarilumab, which interfere with the IL-6 receptor axis, have been disappointing. And it may very well depend both on exactly how you interfere with inflammation and when you interfere with it.
Yeah, and I think someone online, we've had a lot of things online about treatments, and one of the points made by someone online is that we get a lot of hype at first, and we then can feel, and we had a lot about tocilizumab, people saying it's a wonder drug, and then you see the data evolving, and suddenly things don't look so rosy under the cold, hard light of peer review. I'd just like to talk to you now, Kathryn, about the vaccine responses. Have we got the right approach? We seem to every study seems to need more than one vaccine, and I'm just wondering about what that's going to do for us all, and whether or not, so there's two bits which people have asked online, whether or not there is a model whereby a disease produces antibodies, but a vaccine produces better and longer antibodies. So I just wanted to know your thoughts about that.
Sure. So the question of the number of doses is really important for two reasons. One is that, well, if it takes 2 doses to get full immunity, that's more time. So that adds another month for each individual to become immune, or say 3 weeks. But from probably even a bigger issue is manufacturing. So it doubles the amount of vials of vaccine that you need. If you could, so the question is not ruled out yet. I think there may be other vaccines that could be able to do this with one shot. We are working on a platform at our group it's the adenovirus 26, which we showed in some previous data for Zika vaccines, actually had nice durable immunity after a year with one shot. But we, of course, no one actually knows, and we won't know until a year from now, what the durability is for any of these vaccines.
Okay, yeah,
Yeah.
as I said, if the virus doesn't go away, we need something with some durability. Otherwise, we're going to have shots every few months. Yeah, so it's
yeah. I mean, hopefully, it's if we could get it to once a year, that would be great.
Yeah.
The second question that you asked was about whether a vaccine can cause some sort of risk or increased harm to us. And I think it's always important to remember that until you test something in an efficacy trial and in a lot of people, you don't always pick up on all of the safety issues. So, but the question has to do with whether vaccines can elicit some sort of antibody response that once you're exposed to the virus in real life might actually make your symptoms worse. That's not a very common phenomenon. It's known to happen for people who are reinfected with, say, dengue virus. So far, the animal studies for COVID-19 have not shown this phenomenon with vaccines. So animals that get a little bit infected after vaccination don't seem to have worse disease. But this is why we do phase 3 efficacy studies in tens of thousands of people so that we can pick up rare safety events.
Yeah, that's great. So, Anne-Genevieve, I said I would come to you about the cycle threshold. It seems to me that the more cycles you do, the more positives you get, and the less you do, the less you get, and you run a risk if you do less that there'll be people walking around with COVID who you've told haven't got it. How do we sort this one out?
Yes, this is a big issue. I think I have to just to say that I don't know if it was clear, but when we have a high CT, there is a low viral load, and some data have shown that, for example, with some normalized, really quantitative RTPCR, a CT above 37 is corresponding to 1 log viral load, and between 33 and 37 is corresponding to 1 to 2 logs. So even if CT is semi-quantitative, there is a correspondence with the real viral load. And the data I have shown, I think really nicely show that there is a relationship between the CT and the infectivity. So people with this high CT, they are sometimes at the end of the infection, and probably they are not infectious.
So it's really about the interpretation from the laboratory back to the clinicians to say what the significance is. Exactly,
and I really think that we should give some guidelines to interpret this RT-PCR results, and we are working on that.
There was lots of questions on antigen testing, and I wondered if I had a patient with a positive antigen test, what am I going to do different to someone who I get a negative antigen test because of the false negative rate? Dan, they're not for real time, are they?
Well, no. So I think it really depends on the purpose of testing. I think that for diagnostic testing in people who are suspected of having COVID-19, the PCR test is essential because it's the most sensitive test. There have been a number of papers published, mostly based on models, that suggest that if you could have an inexpensive rapid antigen test, even if it is less sensitive, if you can do it frequently enough, the frequency of testing really compensates for the lower sensitivity, particularly since you're missing mostly those people who have very high CT values, as Anne-Genevieve was saying, who are not going to be so infectious. And you would actually prevent more infections by testing more often with a less sensitive test that would also be less expensive.
Yeah.
Okay.
I totally agree with Dan on this.
Yeah. Okay, so my skepticism about them has now been moderated by you guys. Thank you very much indeed. Dan, there's quite a lot about monoclonal antibody treatment. Is there anything there for you to just give us a bottom line on?
There are a lot of studies in progress. The phase 3 studies are really just underway. And they're promising data from non-human primate and other animal model studies. But like so many other things in COVID-19, we really just have to wait for more data.
Yeah.
I just wanted to add too, because we didn't talk about it at all, but something I'm super excited about is monoclonal antibodies for prevention, particularly in post-exposure prophylaxis. So the idea that you might give a monoclonal to someone who has been in contact with their husband or their son, and then they would be potentially protected from infection for the period of time that their contact is infectious. So we don't talk about those programs very much, but I'm really excited to see that data.
Yeah. I suppose, though, one of the issues we have is about the accessibility globally of some of these therapies. So that's why vaccines and maybe cheap drugs are the thrust of science at the moment. I mean, I can understand why we're doing proof of concept things with monoclonal antibodies and with immunoglobulin, et cetera, but I don't know if any of you want to make a comment about their availability globally if we say we come up with a wonderful monoclonal antibody for prevention.
I think that's an important point to keep in mind. That said, a number of the groups developing the monoclonals are trying to determine whether they can also be given by injection or subcutaneously rather than infusion, which would at least simplify their administration, although the cost of manufacture and distribution will remain significant impediments to their more global distribution. I think clearly we need an array of treatments ranging from small molecule to potentially monoclonal antibodies or other more complex and expensive therapies, given the huge global need.
Yeah, I agree. I think of, at least in the prevention space, I think of the monoclonals as a bridging or stopgap measure. So to bridge you until you can be vaccinated, basically. And some of them be given subcutaneously, and you can imagine, say, in a nursing home, their best use case is one person in a nursing home who a staff member is infected, and then you can just go in, get everybody with the injection of the antibody, at the same time, maybe get them all vaccinated, and then protect them for a short period of time. So I think these are all complementary approaches. It's where we need many of them.
Okay, well, we're running out of time as ever. I'd like to first of all thank everybody who's been listening for sending in lots of questions. We've only actually seen a few of those, discussed a few of those, but thank you so much for them. Basically, I think it's been really great update after our break. I think that the diagnostic testing is becoming much more validated, and we're having some exciting ways of maybe being able to test people. I like the point of care and saliva, people being able to do that themselves. So I think that's excellent. The vaccine update, that's really come up logarithmically now, because we've now got some trials going into phase 3 and some data which has been peer reviewed, which is wonderful. Right. That's coming now. And Dan, the therapeutics, well, the remdesivir and corticosteroids were there before, but we're now getting into a phase at which I think we're understanding how to use them and which patients to give them. So I'm looking forward to the next wave of drug therapies that are coming along. I'd like now to wrap up and acknowledge our sponsors for this independent medical education program, Gilead, MSD and Janssen, and for our supporters for advertising this widely on their websites and through their contacts. I'd also like to thank all of you now and ask you to make sure that you complete the webinar survey that's coming up when you close this. And actually, if you want to have materials, more information, please visit us at, and I'm going to do the www.academicmedicaleducation.com plug here. So please do that. And then we're going to come back again in October. And so please join us on Tuesday, the 13th of October in the morning, at lunchtime, or in the evening, wherever you are in the world. And it's for me now to thank all our speakers for a tremendous presentation, for everyone in the back room, and for our program advisors for making this happen. Thank you so much, and I hope you have a good morning, good afternoon, good night. Bye from me.
Thank you.
So long. Thank you, Anton.
Thank you. Bye.